Bueno, en principio la charla la voy a dar en inglés, okay? So, good morning, everybody. Thank you, Izaskun, for the introduction. I'm going to talk about one recent study that we performed in 2019 here in CAP, and that we also recently published. And it's about the microbial communities that grow in a volcano in Antarctica. And that's why we choose the title of A Song of Ice and Fire in the Shetland Islands, Antarctica, Microbial Biomarkers in the Geothermal Band of Cerro Caliente. I guess that most of you know that A Song of Ice and Fire, it's a set of books from George R. Martin. And we think that this title, it's good because in the place that I'm going to talk about, there is a dichotomy between the ice and fire. Also, maybe you know the Game of Thrones TV series that is based in these books. Of course, I'm not going to talk about the fight between kingdoms to get a throne, but the coexistence of different microbial kingdoms in the same place. So I hope you like this story. Let's see if I can pass this one. Yeah. So as I said, this study is published in Astrobiology. So I'm going just to do a summary, but if you are more interested, you can get some details from the article. We are several authors, all from here, from Centro Astrobiología. And we are a mixture between environmentalists, biologists, geologists, chemists, and so on. So you can ask after the talk, but you can also ask any of us here in Centro de Astrobiología. So the place of the study is a geothermal polar environment, which means that is a geothermal site in a polar region. In this case, could be Arctic or Antarctic, but we are going to be focused on Antarctica. And it's a place where there is a transfer of heat from the deep ground to the surface. And this is because of the presence of a magmatic chamber in the ground. And that makes possible, of course, the transfer of heat to the surface. There are some important things in this location that I would like to highlight. There is a combination of snow or ice and also this heat, which makes possible the presence of liquid water, which all of you know that it's essential for life and for the growth of microorganisms. But not only for the growth, but also for the survival, because there is some hypothesis that says that even in Antarctica, during past glaciations, these geothermal sites were a shelter for these microorganisms and some macroinvertebrates. So these microclimate conditions make possible to serve as a refugia for these organisms. Geothermal environments as well, not particularly those located in polar regions, but in general, are possible scenarios for the origin of life on Earth. For the origin of life are in the deep sea, but also there is also an hypothesis of the life emergent in geothermal sites. For example, like this place in Cerro Caliente, for example, that is a geothermal site inland, because there is evaporation of the water, so there is concentration of the simple molecules, that this interaction may favor the synthesis of polymers, and then this synthesis of polymers could make possible the membranous compartments that are necessary for protocells. So that's why these kind of environments are also interesting for the origin of life. But focus on Antarctica, geothermal environments are associated with 4 currently active volcanoes, and one of them, it's an island that is called the Shetland Island. As you can see in the map, it is one of the islands that takes part of the South Shetland archipelago, and it's close to Antarctic Peninsula. The Shetland Island, it's an active stratovolcano. Last eruptions were in the 1967, 69, and 70. So they are very recent eruptions. As you can see in this map, is ring shape because it's actually the upper part of the volcano and the rest of the land is submerged in the sea. And there is a caldera that is actually flooded by the sea water. And in the land, you can find some hydrothermal vents. And some of these places across the island are protected under the Antarctic, especially protected areas. One of the sites that are interesting for us is Cerro Caliente. Cerro Caliente, it's a hill of 107 meters high. So it's a very small hill that in Spanish is called Cerro, that has a narrow line of fumaroles. As you can see in this picture, you can see a different color, like yellow, orange, as compared to both sides, on the right and on the left. This is actually because of the ascending fluids from the ground to the surface. And you can find water, steam, CO2, and also other minor gases. And temperatures can reach 100 degrees. So because of that, because of these high temperatures, there is also a steep geochemical gradient in a very short distance. In fact, if you go just one step on the right or on the left, you will get the permafrost that is cold. And if you stay just in the middle or in the band, you can have higher temperatures. So in general, we can say that this area is snow-free or ice-free. And so there is availability of liquid water, but not only because of the melting snow, but also because some water, it's coming from down to up. So it's water upwelling. And all these conditions make possible the growth of microbial communities here. Of course, these have some similarities with Mars, because in Mars, found some glacial-volcanic features at different latitudes. So you can see, for example, in this picture, that there are some channels that evidences for the presence of liquid water in the past. That one hypothesis is because of the volcanic ice interaction. Of course, may suppose that the ice is melted and then some flowing water may cause these channels. So the microbial communities and the molecules of these communities, that is the DNA, provide a framework for life detection strategies for Mars. If we go back to Antarctica, to the island, we are not only the ones that are interested to know the microbiology in this island. So several studies were already performed. This is a map, the same map as before. I just put some green dots. These are the locations where other studies were performed. It's just a summary because probably there will be more. Well, in fact, there are more studies. So most of them, at least the studies related with microbiology, most of them were performed on sediment or soil. That is, the microbes that were attached to sediment or soil particles. And together with these studies, recent studies has also performed some temperature measurements, trying to associate this possible relationship between the temperature and the microbial community. And for example, this one is from Bendia et al. In 2018, performed in Fumarola Bay and also in Whalers Bay. And they found, as you can see here in this picture, that there is a very steep temperature gradient just in a very short distance. It's just in a matter of meters, there is a difference of 90 Celsius degrees. But despite of these conditions, there were also microalgae, they were detected microalgae and cyanobacteria at different points of the island, and specifically also in Cerro Caliente. Other researchers were also focused on the isolation of thermophilic and cytophilic bacteria, which means that bacteria that lose high temperatures and bacteria that lose low temperatures. And other studies were focused on the submarine sediments of the Port Foster, close to Fumarola Bay. And these studies were focused on archaea. For those that don't know this, exist 3 domains of life. It's bacteria, eukarya, we are eukaryotes, and archaea. Okay, so this study was focused on archaea. And in 2012, in our group, one study was performed there in the Shetland Islands, in a place that is close to the Spanish base. And it was performed in a permafrost to check for the microbial communities that were growing there, and also for the metabolic processes in this cold site. So they conclude that there were some sulfur reduction and ammonia oxidation. But there is a very lack of studies about the microbial composition, the metabolism, and also the geochemistry in science. That's why this is doing some relations, of course, with may show a diverse microbial composition and metabolisms because of this combination of low and high temperatures, but also because of the liquid water availability. So the main objective is to investigate this composition and metabolisms of 3 microbial communities that were located in this site. And so as a specific objective is to check the community structure of bacteria, archaea, and eukarya. And in relation with the metabolisms, we were more focused on the one related with the carbon, specifically the carbon fixation pathways. So in 2011, I was not here, so my colleagues went there to sample some microbial communities that were located at different distance between each other. It was not more than 10 meters. And so they collected some samples that at the time of collection were 88 Celsius degrees, 8, and 2. To answer the question about what's the major carbon fixation pathway of each sample, we performed some carbon stable isotope composition. To answer the question, what's the community composition of each of these samples, we perform massive sequencing of 2 genes. For bacteria and archaea, it is called 16S RNA gene, and for eukarya, 18S RNA genes. And for those that don't know what's this, they have not been lost during the evolution. And they, for sure, I mean, in this sense, they have been accumulating change over time. So with these genes, we can classify the organisms in a sample. And to reinforce or to be more secure about the metabolisms and also the composition, we also perform some assays with antibodies that I will explain later. In parallel with the biological analysis, we also took samples from the substrate to measure some nutrients, the mineralogy, and also my colleagues put thermocouple 2.5 centimeters depth to record the temperature oscillations over time. And here in the bottom of this slide, you can see the appearance of these communities. So the results related with the geochemistry were the following. The ground sample at the time of this collection were at 98 degrees. The pH was neutral, and we identified some nitrate, phosphate, and sulfate. The mineralogy were several silicates, phyllosilicates, and also calcite. And this analysis of the mineralogy, we can conclude that the temperature and the hydrothermal fluids that are ascending from this fault are changing, are altering the original mineralogy of the area. And that's why, I mean, part of the color of this plant is because of that. And also part of the color is because of the presence of microbial communities. The thermocouple that was located in the subsurface at 2.5 centimeters gives this pattern. As you can see, there are some peaks of high and low temperatures over time. The mean, the median, and the mode was between 21, 27. It's okay. It's a warm substrate, but it's not too high and not too low. But there are a lot of peaks. And this means that it was explained because under the ground of Cerro Caliente, the thermal fluids are, of course, moving, and then this transfer of heat is changing. So this temperature gradient is not just spatial, that is in a distance, it's also temporal. And if we get, if we close it, if we did a zoom to this data, we can check that even in a day, there is a high difference of temperature. So for example, here is like 40 degrees. So this is too much. So microbial communities somehow have to deal with these high or low temperature peaks. So we will see what's happening with the metabolisms and also the microbial composition. We perform some analysis of the total organic carbon. Just focus on the first graph. Here we can see that the sample that was at 88 Celsius degrees at the time of collection show higher carbon organic concentration. This could be explained because of the possibly higher temperature of this sample. Because the higher the temperature, the higher the physiological processes, and then the higher biomass. And of course, everything is up to a point, because with 88, it's strange to think like that. But we measured before that the temperature was oscillating over time. So at the end, we can say that the biomass is higher in this sample because of the prevailing temperature over time. So to answer the question about what's the major carbon fixation pathway in the samples, just take a look on the second graph. A carbon fixation pathway means a set of reactions that the microorganisms, well, in general organisms, do to take the CO2 that is in the environment, could be the atmosphere, or the CO2 that is dissolved in an aquatic environment. They fixate from this environment, and they synthesize organic compounds. So to get an idea which is the major metabolic pathway in this sense, we take a look to the stable carbon isotopes. Just stop for a moment just to understand what's that. Isotopes are atoms of the same element that differ in the number of neutrons. So in the case of carbon, there are 3 natural isotopes. Is carbon 12, carbon 13, and carbon 14. In the case of carbon 12, is 6 protons, 6 neutrons. In the case, for example, the carbon 13, 6 protons, 7 neutrons. The nuclear stable, so it's decaying over time. We are not going to talk about this one, but on the stable isotopes. They are present in nature in different proportions. So now, what's the relationship of this carbon isotope with metabolisms? So this relationship is because organisms has, with a specific pathway, has prevalence for one carbon isotope over the other. So for example, the organism that perform the photosynthesis, so has the Calvin cycle, they prefer to take the carbon 12 instead of the carbon 13. Usually we refer this proportion as carbon 13 over carbon 12. And that's why that's the notation, but per mil, that we have in the graph. So with different studies performed by different researchers over time, now we have our reference values for specific pathways. So we can suggest that the values that we have for the colder communities, at least at the time of collection, were related or suggest the relationship with the Calvin cycle. So they have preference for the carbon 12 instead of the carbon 13. That's why it's depleted in carbon 13. In the case of the community at 88 degrees, at the time of collision, of course, it's more related with, suggested to be related with the reverse tricarboxylic acid cycle, which in other words, it's, for those that at least are biologists or environmentalists, is the Krebs cycle, but that goes in the other way around, is in an inverse sense. So we can conclude that the prevailing temperature of each community influence the biomass and also the major carbon fixation pathway. So let's check what's about the community. This is a big graph, but it's divided into three: bacteria, archaea, and eukarya. We can see, but with some exceptions, in the case of archaea, that there is a comparable microbial community composition at the phyloid level between bacteria and eukarya. Of course, in archaea, we have some unclassified and we cannot talk much about it. Moreover, we have to highlight that there are some chlorophyll variant, that is microorganism that has chlorophyll in the microbial community at 88 degrees. But there is some kind, I mean, we highlight this because there is some kind of incongruence with the maximum thermal limit for photosynthesis, that is 33. So, but it could be explained because of these thermal oscillations over time. And in fact, if we take a look to the microbial community, we can see that it's in a gray color and not in a green color that you will expect if you find chlorophyll. So it may think that there are recent fingerprints from this community that we could extract with the DNA. And of course, it explained because of these fluctuations in the temperature, but also we cannot forget that it may be explained because of the water movement, not also because of the thermal fluids that are moving, but also because this is a slope, this is a hill, and then the communities that are in uphill can go slip down to the other communities. So this could not either discarded. If we go closer to this taxonomy, if we talk about orders instead of phyla, we can see that there are some individuals that belongs to some orders that are thermotolerant or thermophiles and are more closer to the community that has 88 degrees. And some cycrotolerants or cycrophiles close to the one at 2 degrees. It makes sense. It could be the reason that there are some thermal niches. But of course, we found some presence of heat and cold tolerant microorganisms in all communities. So for example, some thermophiles at 8 degrees. And once again, it's explained because of this oscillation in the temperature and water movement. We found as well a diversity of metabolisms. Most of them, we were checking all of the individuals, and most of them are aerobic, but some were anaerobic as well. And it makes sense because if you are just taking a sample and you are also taking the parts that are, for example, 1 centimeter depth, probably at this depth, there are also anaerobic organisms. There are photoautotrophs, as we saw before, because there are green algae, there is also cyanobacteria. There is heterotrophs that takes energy from organic compounds, and also chemolithotrophs that takes energy for inorganic compounds. We found also ammonia oxidizers, so archaea, that is ammonia oxidizers, nitrate oxidizers, nitrogen fixers, etc. So there is a microbial composition very heterogeneous and with different thermal traits and very different metabolisms. And now we wanted to check what are the results of the immunoassays, because these results can explain about the composition and also some metabolisms. But before going to explain some results here, just take a look what is the LD chip. Okay, this assay is performed with antibodies. That's why it's called immunoassay. With 2 antibodies, that's why it's called sandwich, and with one of them which has a fluorochrome, that's why it's fluorescent sandwich immunoassay. And these antibodies were, how we collected these antibodies. So we have in the laboratory a set of bacteria, archaea, well, microorganisms that were isolated from different extreme environments, or they were bought from a cultural collection. And with this library of microorganisms, we exposed, what we didn't do it, it's accompanied, we exposed these bacteria to another organism. In this case, it's a rabbit, and the rabbit makes an immune response, the same as we do when we have a virus. And with this immune response, there are antibodies in their blood. So we extract part of this serum and we purify it. And so once we purified these antibodies, we print it in a glass slide that we called, and this device, we call it LD chip, live detector chip. So this live detector chip, at the end, we will be set in a device like this one, in a blue color, and then a sample that will be a soil with an antigen that will be the microorganisms will be passed through. And if the antibody finds this microorganism or part of it, for example, a protein, a sugar, a lipid, or whatever, there will be an interaction. But in this interaction, we cannot measure anything. So we need a second antibody that has a fluorochrome that gives you a signal that you can measure. So these signals will be the red spots that we have here in this picture at the bottom. And so the red spot will mean present, the black absent, but also the difference of intensity can be translated into relative abundance. So at the end, we will have this graph with fluorescent intensity against antibodies, and then it can be translated to a heat map. So this heat map shows also a diverse community. We can find also cyanobacteria that we found with the DNA. So it's at the field level, it's in some cases comparable. And we can find also methanogen archaea, that is methanogenic archaea with this chip. And we can conclude that the microbial communities in Cerro Caliente show heterogeneous microbial composition, and that the use of this chip that can recognize several parts of an organism may serve to determine the bulk microbial composition of environmental samples, where at the end are a mixture of a lot of microorganisms. And in fact, this chip is a core of the solid instrument that it was designed to measure in situ samples, and also with a focus on possible planetary exploration. And just to have some conclusion and to take some ideas from this study, just to say that the geothermal site of Cerro Caliente has a strong temperature fluctuation that must be considered to understand the whole microbial community structure and also the operating metabolisms. These microbial communities are heterogeneous because of the coexistence of individuals with different thermal traits and metabolisms. And of course, because of this coexistence of high and low temperatures. The similarities of Cerro Caliente with glacial-volcanic sites on early Mars confers Cerro Caliente as a relevant site for biomarker studies. And the coexistence of these microorganisms in this site, some of them that are evolutionary distant, because we found archaea, some archaea like aeroarchaea, otamoarchaea, and also green algae that are eukaryotes. So the coexistence of these microorganisms with diverse metabolisms are interesting for designing life detection strategies on Mars. And that's all, and thanks for your attention. And if you have any questions, you can ask now.
Thank you very much, María Ángeles, for the very nice talk. It was really, really nice. Okay, so are there any questions? If you have any questions, you can also write it in the chat, and then I'll read it loud. So if you cannot speak, if you don't have the microphone, then that's something I can do. So maybe I can start with the round of questions. So María Ángeles, you mentioned that you measure the temperature down to a depth of a few centimeters. Is that correct?
2.5 centimeters. 2.5 centimeters.
2.5 centimeters, right. Then for the collection, for the samples that you collected, so you had one at high temperatures and then there are two others at low temperatures. So those temperatures that you measured were at the time of collection, so those temperatures may have changed after the collection. Is that correct?
Well, not after the collection, because once you collect it, you don't know anything, but it means that, I mean, in the sample of the ground, well, close to the sample of the ground, my colleagues put a thermocouple. So if there were thermal oscillations close, just in a matter of centimeters, to the places where they collected the microbial communities, we suggest that these communities have, the spots of where these communities were set have been also fluctuating over time.
Right, okay. So all of them have been suffering all these temperature fluctuations over time.
Yes, so that's why I highlighted that this temperature of 88, 8 and 2 are at the time of collection. It's just like a snapshot, so you cannot guarantee that these temperatures were stable over time. No, it's just the opposite. In geothermal environments are really dynamic and the temperatures fluctuate, but not only on the spatial scale, but also on the temporal scale.
Right, okay, so when you see these differences in the let's say, I mean, I'm not an expert, but when you see, for example, the differences between the 12C content and the 13C, so that means that is changing rapidly with time due to these changes, rapid changes in temperature. Is that correct? Did I get it right?
Yeah, well, temperature has been changing, and the relationship of the C13 and C12, if I understood well, it's at the end a median of the, I mean, it talks about the prevailing temperature of its microbial community, because temperature has been fluctuating. So at the end, this is like a mean. Sorry, not a median, a mean.
So it's like the mean. So it gives you an idea of the mean response, or of the mean,
yes, of the mean value over time that we don't know, because they just took the samples one day and that's all. But we can suggest that the temperature of these microbial communities has been changing over time. The same has been changing just 1 centimeter on the right when we put the thermocouple. So.
Okay, and then the other question that I had was regarding, so you use two methods to characterize the communities.
Yes.
So you have the DNA method, if I understood correctly, and then you have the immune, the
antibodies.
The antibodies, the LD chip.
Yes.
Then I was wondering, so whether there are any differences in the accuracy of using both methods. Are both giving you the same response, or the same results, exactly the same results, or one of the methods is more, you can fine tune better with one method rather than with the other?
Yes. They are not exactly the same, so the results could not be exactly the same, because the DNA, it's an open method, which means that you take a pool of the sequences, the DNA sequences of every organism that was there, and with the LD chip, you are just asking for specific microorganisms, because at the beginning, you have a library in your lab. In our lab, we have a library of microorganisms that is A, B, C, and D. And we expose to the rabbit to these organisms. So we get the antibodies against A, B, C, and D. So it's a closed method. So it could be comparable. We saw that it could be comparable at the phenome level. And of course, we are getting more bacteria, more archaea to enrich this library and be able to detect more microorganisms. But today, we could say that the results were comparable at the phenome level. And even there were some, of course, some differences because one method is open and the other method is closer.
Okay, thank you very much. And then that's the last question. So I was wondering whether on Mars you have any information about these changes on the surface. Has that been measured, or is that something that will be measured soon in one of the future missions going to Mars?
So I cannot answer this question because, well, I know that there are some glacio-volcanic environments. I know as well that the geothermal activity, I think the geothermal activity in Mars right now is not detected, but maybe I could be wrong. I'm just talking about ancient Mars. I mean, in the past, there were some volcanic ice interactions because in the past, they were active, and that's why they could, if life was developed, there could be some biomarkers there, and that is the context of this study that we perform in Cerro Caliente. But so far, I mean, I cannot answer properly this question. As far as I know, there is just no geothermal activity, if I'm not wrong.
Okay, thank you very much. So are there any other questions? No?
Is everything clear?
Yes. Maybe I asked too many questions. No other questions? Okay, so I think this is it. Well, thank you very much, María Ángeles. It was a very nice talk. And thank you very much for doing it in English. Thank you very much. It was really, really nice. Okay, and then, bueno, yo creo que ya puedo cambiar a español. Pues la próxima semana tendremos otro seminario. Muchas gracias, María Ángeles, ha sido muy interesante. Y la próxima semana también tenemos otro seminario. Creo que el de la próxima semana será el de Gonzalo González Abad, que se encuentra en el Center for Astrophysics. Y bueno, enviaré la información del seminario pues a principios de la semana que viene. Bueno, muchas gracias a todos por uniros hoy. Vamos a ver cuál ha sido el problema con la conexión de Zoom desde el CAP, a ver si lo podemos solucionar para la semana que viene. Y bueno, seguimos improvisando, haciendo lo que podemos sobre la marcha. Y bueno, pues muchas gracias y hasta la semana que viene.
Vale.
Hasta luego, muchas gracias, adiós.